Welcome, class. Today in agricultural biology, we're going to be setting up a lab. I'm going to show you how to put up all of these seeds in various containers and how to use the Spark data logger so that you can generate information on CO2 gas as well as oxygen gas. We've been studying cellular respiration, and this particular lab is going to be measuring the O2 as well as the CO2 and see what role they play. We're going to answer basically two questions. How does the rate of cellular respiration, how does it impacted or affected by temperature? And the second one is, do dormant seeds produce more or less CO2 during the germination process through soak seed? So we're looking at those two questions, answering those through our lab today. So let's go ahead and get started, and let's talk about the things you're going to need to kind of set up the lab. First of all, you're going to need to get one of these data loggers, the Spark data logger. It'll allow us to not only collect data, but also do our lab write-ups right on this data logger. So you'll need that. We're not going to use any paper. It's not going to be a paper lab. It's going to be all digital right on the data logger here. We're also going to need 3 different groups of pea seeds. You're going to need dormant seeds. You're going to need seeds that have been soaking in water at room temperature for the last 24 hours. And of course, you're going to need some seeds that have been in ice for 24 hours as well. So those are the 3 treatments that we're going to be looking at, and we're going to see how the cellular process moves forward based on temperature and what gas is given off, oxygen or CO2. So to get things started, we're going to first of all start with the dry seeds. These are just regular pea seeds. There's about 25 of them in. You only need 10 really, but you're going to put in 10 of them. And what I'm going to do is I'm going to use this bottle here with a funnel and just pour the dry seeds in there. I'm going to count out 10. Put that off the side. I'm going to put out 10. And they don't get any water or anything. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And I'm going to set that off to the side. Put these back in. Now what we're going to do is, on the first run now, we're going to split the class in half. Half of you are going to put in a CO2 meter. The other half of you are going to be putting in a sensor that measures oxygen. So this is what the CO2 sensor looks like. And you're going to just press this calibration button until it starts to, there we go, blink. Well, first of all, plug this in right to our data logger. Then we're going to calibrate it. Now, once it's plugged into the USB port, it's going to come up blinking. I'm going to hold it down for about 3 seconds till the light stops blinking. And that's going to be, I'm going to let that calibrate for about a minute. When that thing is done calibrating, it'll blink again. And the surrounding, the surrounding atmosphere should be anywhere from 300 to 400 parts per million. So once that's done, we're going to put that right in here like that. And then on your Spark data logger, you're going to go to the data collection screen. And for this particular lab, it's going to be on page number 13. That's where you collect the dry or dormant seeds. So when you finish that, then go ahead and hit play, and you're going to let that collect data for 10 minutes. While that's kind of cooking and collecting data, you can start getting your next sample ready. This one is going to call for water, room temperature water. So you have to pour off some of the water in here. So while this is being collected, you're just going to pour the water down here to separate it. And then you're going to collect let them kind of drain it a little bit. Now remember, you're letting the dormant dry seeds stay in this bottle, this capture bottle, this collection bottle, and you're going to be sampling the amount of CO2 it produces for the next, right now, about 9 minutes. Well, that drains off. Let's head off to the side. And then the last one is going to be, of course, the one from the ice water. Oh, there you go. I'll put that in there. Always good to have a few washcloths around as well. So let's say the 10 minutes is up. You're going to collect the data. set this off to the side. You're going to clean that up later. You're going to rinse out the bottle, and then you can dry it, and you can use it for other things. So in this particular case, we're going to do the same thing again. We're going to pour out exactly 10. That's 6, 7, 8, 9, and 10. And then again, the same thing. Then you're going to go to the next page, is page 15. And this is going to, you're going to be collecting data for now that the seeds that have been soaking in water at room temperature for the last 24 hours. That soaking, is that going to help the process? Is it going to hinder it? And what kind of CO2 emissions is it going to have? Because if germination is going on, then there are enzymes in the seeds that are beginning the process of breaking down the carbon dioxide that's been stored up in the seeds and should be releasing CO2. But is it going to have more CO2 or less CO2 based on the temperature? So you're going to let that run for 10 minutes. And again, set that aside. As you are waiting for that to run, you're going to get your other collection bottle. And this collection bottle, you're going to be getting out the cold seeds. Again, same thing. They've been put in water for 24 hours. Let's set that over here for now. Put that in the wrong place. All right. So we pour it off, and we're going to collect all the pea seeds. And by the way, you don't have to use pea seeds. You just have to use a dicot. That usually works the best. And we're using some pea seeds today. You could use fava beans. You could use beans. Either way. And we'll count 10. 1, 2, 3, 4, 5, 5, 6, 7, 8, 9, and 10. All right, those are nice and cold. And before the cap that off so it doesn't get too acclimated to the surroundings. Now let's say the 10 minutes is up, so I stop it. Now I have basically 2 runs. The first run was with dry, dormant seed. The second run was with the seed that has been soaking in room temperature. And the last run is going to be the temperature, the colder temperature. Put that right in there. You could actually do 2 of these at a time if you like. And by the way, we're going to go up to the next graph. And that graph is on page 18 of your data logger. I'm going to hit play. And now I'm going to let that go for 10 minutes. So you could actually be doing 2 at the same time. But again, what I'm trying to do here is I'm just trying to demonstrate to you all the 3 different treatments you're going to do to the seeds. And because of that, it might be easier for you to kind of keep it, do the dry dormant ones, and do the ones at room temperature that have been soaking in water for 24 hours, and then the cold ones that we're soaking for 24 hours. So that way you can kind of keep it straight. But if you think you can handle 2 at a time, it'll save you basically 10 minutes, and you'll probably be able to get it done on time. So once we have that data collected, then what you can do is, while it's collecting data, you can go ahead and fill out the charts that have to do, like for instance in this one, it talks about the initial CO2 level to the final CO2, so you can see the change. And again, you're going to have one graph with 3 different runs. First being dry dormant. Number 2 is going to be the one that's been soaking in water at room temperature for 24 hours, and then the one that's been soaking in cold water for 24 hours. And you can go through that. Remember, the 2 questions we need to answer: what does the temperature, how does that affect its respiration rate? And number 2, what would be the amount of CO2 being produced? Will it be more in the dormant one, or the one in the cold one, or one in the room temperature one? That's for you to figure out. And I hope this helped. And like I said, it shouldn't be too hard for everybody to do this lab within the time frame that we've got. So if you have any questions, don't forget to ask, and we'll see you back in the lab. Thanks.